Thank you very much. And of course, thank you very much also to you, Nikolai, for this very nice introduction. And of course, also to the whole Evosep team. It's always a pleasure to not only give presentation for you, but then also to work with you. And maybe also for the audience, it's already like a small disclaimer from my side, but Nikolai also stated, so we're working already together for many years and have some very exciting projects done over the past. Maybe also a second disclaimer from my side before I'm starting. So as Nikolai said, I'm working at Omega Diagnostics. I'm also co-founder together with Sophia Doll and Sebastian Ferreira-Winter and also Ole Worm of this company. And just to give you a bit an overview of the company, so we see ourselves, or we are a small tech startup, and our big mission is to improve health by proteomics. And internally, we have a huge focus on automation of the proteomics pipeline, but then also on software solution. And here we also apply some open source software solution where we want to give something back to the community. But then, of course, we also have some proprietary projects running. And of the open source software, it's also like a small advertisement now. Please check out the two that you can see here, like OmicLearn and AlphaPet. And OmicLearn, it's like a machine learning tool for your biological data. So you can use their machine learning very easily for binary classification, for example, case versus control comparison, and get some nice machine learning models out of this. And it's open source, as I said, so you can just go to GitHub and you can also contribute if you want. And please let us also know if you find some bugs or if you want to contribute. It's, as said, it should be like a community approach. And the same goes for AlphaPet, where we are contributing together with Matthias Mansleb. Yeah, but now let's start with the talk. So first of all, I want to emphasize why we have to go to automation, and this is because, in general, because we have a need for high throughput, especially in plasma proteomics, and this is where I'm spending most of my time. So of course also in other clinical samples, but I have a big interest in plasma proteome. And I think it's quite clear why we should analyze the plasma proteome. And this is because the blood and therefore also its liquid component, the plasma, are flowing through all of our body. And on its way through our body, it can collect all kinds of information in the form of molecules like proteins, and they are then a reflection of our health and disease state. And this, of course, also the case in the clinic and not just pure fantasy. So this is from a survey from our close collaboration partners at the Klinikum Großharden here in Munich. And according to them, 77% of all clinical decisions, they are based on some kind of a lab test. And it is very reassuring to see actually that, that proteins have the largest share of the analytes that are measured in the clinic. And as you can see also on this slide, so we have here 9 million lab tests. And of course, if you simply want to handle such a thing in the clinic, you need, of course, automation. And this is also the reason why processes in the clinic are automated. Another reason, apart from this throughput reason, is that you, of course, reduce manual sampling errors. And that's, of course, also a big advantage. But this, of course, direct clinical application. Most of us will work actually with a different field of application. This is more in biomarker discovery. And there's also a reason why we want to work there. And the reason is that we don't have biomarkers for all diseases. And most biomarkers that we have, they are actually quite old. So we really need new biomarkers and find new biomarkers. And one reason why we don't have new biomarkers is often said is because the plasma proteome has such a high dynamic range problem. I think this is true, that's one of the reasons, but maybe it's not even the main reason. So I think the main reason is actually biological variation, and then of course also missing knowledge that we are having. What do I mean with biological variation? So here on this slide you see now just technical variation. This is like one plasma sample prepared on several days, and then I calculated for all the proteins the coefficient of variation. And then you see that the majority of proteins, like 71%, can be quantified with a CV below 20%, and CVs below 20%, they're already quite good. So our proteomics workflows, they can actually work nicely and reproducible. We can include now also some biological variation. So this is now intra-individual variability. This means I prick myself several times a day for several days in my fingers, and then I calculate the CVs. And still you see the majority of all proteins, they have low CVs. So the plasma proteome is quite stable within a person. However, the issue appears if we have different individuals and calculate now the inter-individual variability, and then you see just a small number of proteins can be stable quantified across individuals. And what does this mean? That we have a low intra-individual variability, but a high inter-individual variability. This means that we really have to go to large-scale cohorts. For example, for typical case and control comparisons, there we need large-scale cohorts. We need really a lot of individuals to compare to each other. Or as an alternative, we also can use this fact that the proteome is quite stable within a person over time. So we can use some longitudinal sample sets. But then again, you would have several samples per person, and then you are ending up with, again, a quite large-scale cohort. And we summarized these large-scale cohorts a bit under the name rectangular strategy and for biomarker discovery. And what you can see here on the slide now is first the classical approach, which we termed then also in contrast to rectangular strategy, the triangular strategy. And here you typically start with a low number of individuals. You try to profile them with shotgun proteomics in as big depths as you can. Then you end up with some candidates, go to the next step, try to verify them with targeted proteomics, and the surviving candidate should be then validated again in a much larger cohort. I think this has a quite low likelihood to succeed because of the high biological variation in the first step. So you will end up, due to these small numbers of samples, with unlikely candidates. And then you go through a process with 3 different technologies. So that's, I think, quite hard to do. An alternative approach could be this rectangular strategy, where you try to go instantly for several large-scale cohorts and analyze them in parallel. And then you just compare always the significant outliers across the 2 cohorts, and these are then your true biomarker candidates. Which is also nice here is that you just would use now one technology that you can master. And actually, we are also, of course, we're not coming up with this idea. We just were looking to genomics, and they're using Jiva study. That's more or less the same, I would say. The biggest disadvantage of the rectangular strategy, also to be open about this, is that you need large-scale cohorts, and you need, in this case, 2 large-scale cohorts, and it's always hard to get a lot of samples, I would say, from collaboration partners. So that's definitely a drawback. If you have these large-scale cohorts, you also want to analyze them, of course, and here automation helps you a lot. And if you look in our MS-based proteomics workflow from sample preparation, LC-MS, spectral interpretation, and then also data analysis, I think what you can automate is actually all of the steps. I mean, we all know that the LC and the MS, or the combination of both, it's already more or less automated. And I think we also see more and more publication about automation of sample preparation. And we use automated workflows, so back in Matthias Mann's lab for quite some time, and also here at Omic Era, we're using some robotic systems. And I also wanted to comment, like, from my own experience about this. So this is just lab automation now. And there are, of course, various vendors, and all of the typical vendors, especially the big ones, they will have fantastic instruments. So I think all of them are working nicely, and at least from Agilent, Hamilton, and Tecan, I know labs that are working with these instruments, and all of them, they were quite happy. Of course, these 3 vendors and their robots, they will be quite expensive, and a more low budget solution might be like an open Tron system, and you can also very nicely automate your systems with it. We're also using in-house open Tron systems, but then it's more for repetitive work and not directly for sample preparation. What we use for sample preparation is actually like an Agilent Bravo system. The reason is, for us at least, is the high throughput that you can get quite easily. So it's working with a 96 wellhead. We also can shift to a 384 well head, and then you can prepare a lot of samples in parallel. The instrument itself is also highly reproducible. It's also very flexible. And just in the case you're looking for an instrument, it also has a very easy to use user interface, and you can get almost started instantly with the system. So that's like the system that you always also see on my slides. What I mean with the flexibility is you can implement various workflows on it. and with also various grades of automation. So for example, you could just exchange a part of this robot, it's called the head, that's this part actually, and then you could do, for example, phosphor enrichment or antibody enrichment, so whatever you want, you can enrich with such an head. And you can also look this up. This, for example, a publication from Albert Hecht's lab. Maybe also a comment on automation. So semi-automation is always very easy to achieve, and fully automation is usually a bit more tedious to achieve, I would say, and more cost intensive. And quite often, I think for most of the processes that you will need in a lab, semi-automation is usually good enough, I would say. Yeah, maybe also mention the Evosep system again, as Nikolai said. So I had the honor to work very early on with the Evosep system. This is just also to show you how far you can go with automation. So even some years ago with the first system, we were running like 2,000 samples in a row. And this was still in the development process of the instrument. They were lining up already some technological inventions, so to say, and replace some parts, and then you see really how reproducible the instrument was going to be. So at the beginning we had quite some errors, then they exchanged, for example, a needle, then the error rate dropped instantly, and then they optimized like a seal, and then from then on actually the Evosep system, and this is really the first breadboard model, was then running without any issues. And I think what's always very stunning is like this figure C down here, which you will also find in the paper. It is like the pressure increase of the column, like the back pressure of the column. So you see the runs 1 to 3, and here the runs, so the last 3 runs, and you see this, the pressure is almost not increasing. And this also helps us in our hands, at least, to run thousands of samples without any issues. And I also want to show you one of the studies, so just as an application example. We applied it. So this is a study we just published some months ago together with our collaboration partners here at the Klinikum Großhadern. And the idea of the study was to get a biological insight into COVID-19 and to see also which proteins are changing COVID-19. So we didn't have the intention to find new biomarkers in these studies. Nevertheless, the reviewers made us to do such comparisons. Some of the technical notes you will also find in this app note, which we published together with Bruker. So please also look this one up. Maybe just very quickly how the study was built. So it's actually a quite nice study. We had very nice controls here. They were actually all like these patients were all suspected to have COVID, so they were coming to the hospital with COVID-19-like symptoms, but they were then PCR negative. And we also had like 31 COVID patients with the longitudinal trajectories. And there are now some quite nice aspects of the study. The one is we have longitudinal trajectories, and the other is we have very good controls. What's not so nice is, for example, if you would now do a comparison between the COVID-19 cases and the controls, because it's not very well balanced, I would say. But with this longitudinal trajectory, you can then go back and also check such changes between the two groups, and then you can confirm them. So in this study, we measured around 720 samples. And I think what you can easily do is then, for example, for this longitudinal sample set, you can now ask, so what proteins are changing? And in here, actually, we saw that a quarter of all the proteins that we quantified were significantly changing over time. And then you also can make some of these nice longitudinal trajectories. And I like them always a lot because they show you quite a lot of biological insight. So for example, that's now the median protein level of different proteins that is significantly altered over time. And that's now the timeline for these individuals. And then you can cluster them together just with hierarchical clustering. And then you would see, okay, this first cluster is dominated by proteins of the innate immune system, for example, CRP, SL1, CD14. And these patients, they were coming in the hospital, they were very, very sick. And then, of course, they're getting healthy over time, and then their level of the inflammation proteins go down. The second cluster was dominated by proteins of the coagulation system. These are increasing after hospitalization when the patients are feeling better. And that's interesting because problems with the coagulation systems, they're one of the main issues for complications in COVID-19. And the last cluster is dominated by immunoglobulins, and typically that's not so interesting. But if you look up the paper, that's actually the most exciting part in the paper, because you really see on an individual-specific level how the immunoglobulins are increasing, so the antibody response to COVID-19. So in the, as I said, the reviewers here also made us to do some comparisons, not only between cases and controls, which we also would have done by ourselves, but then also to say, okay, if a patient is, for example, has a bad outcome in the future. And I think for this, the study was a bit underpowered, but we also measured now a new study, and here you can ask exactly such questions. So the previous study had 720 samples, the new study has now almost 2,400 samples, and then you can ask complete different questions. So you could here also use heavily machine learning, for example, binary classification, and just predict if a sample is now from a COVID patient or from a control. And as you can see, so just from the plasma proteome, you can say with a 90% probability that a sample from a COVID patient is from a COVID patient, and a sample from a control is from a control. And of course, in COVID, we want to have 100%, but I think it's already quite stunning that you can distinguish cases and controls that good. You also see here the typical ROC curves that you will see. And there's also a nice take-home message. So we have here quite a lot of clinical parameters, so more than 300 different ones. And you can also put them in some machine learning models and predict if a sample is from a COVID-19 patient or control. And this works actually fairly nicely, but the proteomics data, they completely outperform all the clinical data that we had here. What's also nice, you can maybe ask if the model is really working. For example, if a patient comes at another day at the hospital, like 5 days later, if you still can distinguish case and controls. And here the answer is yes, you can do this. So we just took then the samples on day 5, and we got actually the same result as at day 0. So you can really distinguish case and controls. You can also ask different questions, for example, about treatment. Does a patient that comes in at the first day of hospitalization, does he need now a distinct treatment? For example, oxygen, which is this CPAP. And then you can actually predict, okay, this patient will need in the future CPAP. This is also possible, of course, with an ECMO, so with an artificial lung. You also can predict outcome. So is my patient dying maybe in some days? And this you also can predict. And I think what's very stunning is that you can even predict some timelines. I mean, this doesn't work 100%, but I think it's quite nicely. So you can also predict the day of the event. And typically, so in this case, the patient were dying. If a patient was dying, the patient was dying like 25 days after hospitalization, and we typically can predict it with an error of 4 days with these proteomics data. So I think that's quite cool what you can do here. But of course, the real game changer is then also if you can predict when a patient needs a distinct treatment. For example, my patient goes now in 5 days on the intensive care unit, so you can also maybe plan a bit better in the hospital. So this is actually where it should go in the end, and we are also working on some machine learning models to really predict such events. Yeah, with this, I'm actually already done. So maybe a short summary. So I was talking a bit about automation, and of course I was also saying that I'm quite convinced that it's possible to find new biomarkers, but I'm also quite sure we have to switch to large-scale cohorts with this. And what I didn't mention, but I want to emphasize it here, is always use quality controls for plasma proteomics or serum proteomics. And as you saw on the last slide, it's possible to get some medically relevant information by plasma proteomics for COVID-19 patients. Yeah, with this, actually, I want to say thank you for your attention. I'm also very happy to get some questions. And as said before, I'm also very happy if you have some feedback for our machine learning tool, OmicLearn, and of course also for AlphaPept. And a lot of thanks goes, of course, to all the collaboration partners that you can see on the slide. Yeah, with this, I'm actually finished. So thank you very much.